This presentation is an introduction to urinalysis, Chapter 2 of the reference book, Urinalysis and Body Fluids, 6th edition by Susan King, Strassinger, and Marjorie Schwab de Lorenzo. The audience for this presentation is students in a medical laboratory technician or medical laboratory scientist program. This presentation includes a lecture, definitions of terms, and some questions over the objectives. My name is Jean Riddell, and I hope you find this presentation useful to your studies. The objectives for this presentation include being able to list 3 major organic and 3 major inorganic chemical constituents of urine, describe a method for determining whether a questionable fluid is urine, recognize normal and abnormal daily urine volumes, describe the characteristics of the recommended urine specimen containers, describe the correct methodology for labeling urine specimens, state 4 possible reasons a laboratory would reject a urine specimen, list 10 changes that may take place in a urine specimen that remains at room temperature for more than 2 hours. Objectives continued. Discuss the actions of bacteria on an unpreserved urine specimen. Briefly discuss 5 methods for preserving urine specimens, including their advantages and disadvantages. Instruct a patient in the correct procedure for collecting the following specimens: random, first morning, 24-hour timed, catheterized, midstream clean catch, suprapubic aspiration, 3-glass collection, and pediatric. Identifying a diagnostic use for each collection technique. Describe the type of specimen needed for optimal results when a specific urinalysis procedure is requested. This slide lists key terms that you should understand the meaning of as a laboratory technician or scientist. These terms are albumin, urea, anuria, catheterized specimen, chain of custody, first morning specimen, midstream clean catch specimen, nocturia, oliguria, polyuria, suprapubic aspiration, and timed specimen. History and importance of urinalysis. The 3 parts of urinalysis are physical, chemical, and microscopic examination. Throughout history, we can tell by the documentation in images, drawings, and records that physicians or healers made observations of urine to determine the cause of a person's health issues. The healers knew that certain characteristics in urine might give a clue to what is happening, even if they didn't understand the science. They observed the color, clarity, volume, odor, viscosity, or sweetness. If a urine attracted ants or tasted sweet, they would assume that there was sugar present in the urine. They didn't know what diabetes was, but in a way, they were detecting the excess urine sugar caused by diabetes. In 1627, Thomas Bryant wrote a book about disreputable scoundrels called piss prophets, who charged large fees to perform the urinalysis, even though they had no training or understanding of urinalysis. So people who did bad urinalysis testing caused England to pass the first medical licensure law. When the microscope was invented in the 17th century, this expanded what could be learned from urine. Urine is readily available, and it contains a lot of information about medical processes, which led to the expansion of tests as science developed. When the testing became more time-consuming, the physician dropped it as a routine test. However, as science developed our modern methods, we have been able to find more ways to use different urine tests routinely in the laboratory. The Clinical and Laboratory Standards Institute, or CLSI, defines urinalysis as the testing of urine with procedures commonly performed in an expeditious, reliable, accurate, safe, and cost-effective manner. Reasons for performing urinalysis identified by CLSI include aiding in the diagnosis of disease, screening asymptomatic populations for undetected disorders, and monitoring the progress of disease and the effectiveness of therapy. The formation of urine. The kidney filters 170,000 milliliters of plasma a day, but only about 1,200 milliliters becomes urine. The tubules of the nephron reabsorb water and vital nutrients such as proteins and glucose. If you notice that urine is yellow and that plasma is yellow, this makes sense because urine is formed by the filtration of blood. Urine is formed from an ultrafiltrate of plasma. Urine composition. Urine is composed of about 95% water and 5% solutes. These solutes include urea as well as other organic and inorganic compounds. The composition of urine is affected by what you eat, how much activity you have, metabolism, and endocrine functions. Urine composition. Urea. Urea is an organic waste product produced in the liver by the breakdown of proteins and amino acids. Urea composes 50% of dissolved solutes in urine. Urine composition and other organic substances. Other organic substances found in urine are creatinine and uric acid. If you aren't sure the sample is urine, you can test it for creatinine and urea, as both substances are present in much higher concentrations in the urine than in other body fluids. Urine composition, inorganic substances. Chloride is the major inorganic solute found in urine, followed by sodium and potassium. Other inorganic compounds are found in trace amounts. The range of these inorganic compounds are greatly affected by diet, so a normal range cannot be established. Urine composition, other constituents. Other urine constituents include hormones, vitamins, and medications. Formed elements such as cells, casts, crystals, mucus, and bacteria can become part of the urine, but they are not part of the original filtrate and enter the urine from other parts of the urinary tract as urine moves through the kidney, bladder, or urethra. When there is an increased amount of formed elements, this can be an indication that a disease state is present. Urine volume. The volume of urine that a person excretes each day is dependent on their state of hydration. The normal range for an adult is 600 to 2,000 milliliters per day, but the usual range for adults is 1,200 to 1,500 milliliters per day. Abnormal urine volume terms include oliguria, which is a decrease in the volume below 400 milliliters per day and is usually caused by dehydration. Anuria is a cessation of urination and is usually caused by kidney damage or a decrease in blood flow to the kidneys through the renal artery. Polyuria is an increase in the amount of urine above 2,500 milliliters per day and can be caused by disease states such as diabetes mellitus and diabetes insipidus, or it can be caused by caffeine, diuretics, or alcohol, which inhibit antidiuretic hormone. People with diabetes mellitus and diabetes insipidus will have an increased thirst or polydipsia. We can differentiate disease states caused by polyuria. A urinalysis can differentiate polydipsia caused by diabetes insipidus from diabetes mellitus. If a person has increased thirst or polydipsia, they will usually have polyuria. If we test the specific gravity and it is low, this is due to low levels of antidiuretic hormone, ADH, or a decrease in the function of ADH. This is diabetes insipidus. When a person has polydipsia and polyuria and the specific gravity is high due to glucose in the urine, this is caused by low insulin or insulin resistance in diabetes mellitus. High levels of glucose in the blood lead to the nephrons not being able to reabsorb all the glucose. This leads to the loss of glucose in the urine. The extra glucose in the urine pulls more water into the urine, which increases the urine volume. Specimen collection. When handling a urine sample, you should observe standard precautions. Gloves and a lab coat must be worn. Specimen collection. The specimen collection container should be clean, clear, dry, wide mouth, flat bottom, and leak-proof containers. Disposable single-use is preferred. If a patient uses a container from home that has not been cleaned well or has residual cleaning solution in them, it can lead to invalid test results. Single, sterile, wrapped collection containers should be used when a microscopic culture is ordered. Urine can be transferred to a conical tube for centrifugation and microscopic examination. Specimen collection, labeling. When a urine sample is labeled, it must have two identifiers, such as full name and date of birth. The time and date of collection must be on the label. The label should be placed on the container and not the lid. The lid may have an extra label, but not the only label. The sample type should be on the label: random urine, midstream clean catch, catheterized, or suprapubic aspiration. Urine cups may be used for other samples that are not urine, therefore it is important that the sample type is listed. Specimen collection, requisitions, or orders. Each sample should have a requisition for the tests that are ordered. The information on the requisition must match the patient ID on the label. The type of specimen and means of collection should be listed. If the patient is taking any medications that may interfere with the testing, these may be on the requisition. The time the specimen is received in the laboratory should be recorded on the requisition. The electronic requisitions usually allow staff to receive the samples electronically. Specimen handling. Just because a sample is not difficult to obtain doesn't mean that we should be lax in managing the urine samples. Changes do happen both in the body and in the sample in the specimen container. Because many changes can happen in a sample after it's collected, we should complete the urinalysis as soon as possible or within 2 hours of collection. The next two slides show you the major changes that can occur while urine is left to sit at room temperature for prolonged periods of time. The most common preservation is refrigeration, and it is very important for culture samples which aren't plated right away to prevent overgrowth of bacteria in the sample and a false positive for urinary tract infection. Chemical analysis should be done at room temperature samples. So if you did refrigerate a sample, you would need to warm it to room temperature before performing testing. Specimen handling, preservatives. The most common form of preservation used in the laboratory is refrigeration, which does not interfere with chemical tests, but it can precipitate amorphous phosphates and urates. It prevents overgrowth of bacteria in the sample. Boric acid prevents bacterial growth and metabolism, but interferes with drug and hormone analysis. It is good for maintaining an acid pH and for transporting samples. Formalin is an excellent sediment preservative, but because it is a reducing agent, it interferes with chemical tests for glucose, blood, leukocyte esterase, and copper reduction. Sodium fluoride is a good preservative for drug analysis, but it inhibits reagent strip tests for glucose, blood, and leukocytes. Changes to unpreserved urine after 2 hours, slide 1 of 2. A urinalysis should be performed as soon as possible or within 2 hours of collection. Samples that cannot be tested within this time period should be refrigerated. This chart shows the changes that occur in an unpreserved urine if it is left to sit at room temperature for more than 2 hours. The color will darken due to oxidation or reduction of metabolites. The sample will become less clear or cloudier due to bacterial growth and precipitation of amorphous crystals. The odor of the urine will increase due to multiplication of bacteria and the breakdown of urea to ammonia. The normal pH is acidic, and the pH will increase due to the breakdown of urea to ammonia by urease, producing bacteria or loss of carbon dioxide. If glucose is present, it will decrease due to bacteria utilizing it for glycolysis. Ketones will decrease due to volatilization and bacterial metabolism. Changes to unpreserved urine after 2 hours, slide 2 of 2. This slide shows additional changes to unpreserved urine samples at room temperature. If bilirubin is present, it will decrease due to exposure to light or photooxidation to biliverdin. Urobilinogen will decrease due to oxidation to urobilin. Nitrate will increase due to multiplication of nitrate, reducing gram-negative bacteria. Red and white blood cells, as well as CAS, will decrease due to disintegration in dilute alkaline urine. Bacteria will increase due to multiplication. Trichomonas will decrease due to loss of motility and death. This shows you why it is important to do the testing within 2 hours of collection. Specimen rejection. Improperly labeled and collected specimens should be rejected by the laboratory, and appropriate personnel should be notified to collect a new specimen. Unacceptable situations include specimens in unlabeled containers, non-matching labels and requisition forms, specimens contaminated with feces or toilet water, containers with contaminated exteriors, specimens of insufficient quantity, and specimens that have been improperly stored or transported. Your laboratory will have criteria for rejection in your standard operating procedures, which you should follow. Specimen types. The random urine sample is the most commonly collected sample. The sample can be done at the convenience of the patient and is used for routine screening. The first morning urine sample is the ideal sample because it is more concentrated. This is the best for pregnancy tests to prevent false negative results. This is also ideal for orthostatic proteinuria. Patients should be instructed to collect the first sample when they wake up and deliver it to the laboratory within 2 hours or refrigerate the sample. The 24-hour sample is used for precise measurements. Some solutes exhibit diurnal variation, such as catecholamines, 17-hydroxysteroids, and electrolytes, in which the lowest concentration is in the early morning and the highest concentration occurs in the afternoon. The 24-hour collection must begin and end on an empty bladder. We want all the urine produced during the time period. Therefore, the urine at the start of the 24 hours is discarded, and then the urine at 24 hours is added to the container. These samples must be kept on ice throughout the collection, and some require specific preservatives solutions. The patient usually is instructed to pee into a urine collection hat and then transfer it into the container so they can avoid contact with the preservative. The 24-hour sample must be well mixed, measured, and then an aliquot is taken to be tested. The catheterized specimen is collected under sterile conditions by passing a hollow tube catheter through the urethra and into the bladder. The catheterized specimen is excellent for bacterial cultures because it does not have the contamination from the urethra. This is performed by a physician or a nurse, not by laboratory staff. The midstream clean catch specimen provides a safer, less traumatic method for obtaining urine for bacterial culture and routine urinalysis. It provides a specimen that is less contaminated by epithelial cells and bacteria and is therefore more representative of the actual urine than the routinely voided specimen. The suprapubic aspiration is obtained by passing a needle through the abdomen into the bladder, making this good for cytology and culture. This can be used to remove urine from a patient with a blocked urethra due to stone or prostate issues. The 3-glass collection method is performed by the urologist for the detection of prostate infection. Cultures. When both a routine urinalysis and a culture are requested on a catheterized specimen or midstream collection, the culture should be performed first to prevent contamination of the specimen. A collection transfer kit can also be used. Pediatric specimens. Collection of pediatric specimens can present a challenge. Soft, clear plastic bags with hypoallergenic skin adhesive to attach to the genital area of both boys or girls are available for collecting routine specimens. Sterile specimens may be obtained by catheterization or by suprapubic aspiration. Care must be taken not to touch the inside of the bag when applying it. The anal area should not be inside the bag to avoid fecal contamination. Drug specimens. Urine specimen collection is the most vulnerable part of a drug testing program. Correct collection procedures and documentation are necessary to ensure that the results are those of the specific individual submitting the sample. The chain of custody is the process that provides this documentation of proper sample identification from the time of collection to the receipt of laboratory results. The chain of custody is a standardized form that must document and accompany every step of drug testing, from collector to courier to laboratory to medical review officer to employer. For urine specimens to withstand legal scrutiny, it is necessary to prove that no tampering of the specimen occurred, such as submission, alteration, or dilution. All personnel handling the specimen must be noted. The specimen must be handled securely with a guarantee that no unauthorized access to the specimen was possible. Proper identification of the individual whose information is indicated on the label is required. Either photo identification or positive identification by an employer representative with photo ID is acceptable. Urine specimen collections may be witnessed or unwitnessed. The decision to obtain a witness collection is indicated when it is suspected that the donor may alter or substitute the specimen, or it is the policy of the client ordering the test. If a witnessed specimen collection is ordered, a same-gender collector will observe the collection of 30 to 45 milliliters of urine. Witnessed and unwitnessed collection should be immediately handed to the collector. If the urine temperature is not at body temperature, or if the sample is an unusual color, it may indicate tampering. Definitions and terms. Albuminuria. This is protein or albumin in the urine. Anuria. Complete stoppage of urine flow. Catheterized specimen. A urine specimen collected by passing a sterile tube, a catheter, through the urethra and into the bladder. Chain of custody, or COC, is a step-by-step documentation of the handling and testing of legal specimens. First morning specimen is the first voided urine specimen collected immediately upon arising from sleep. It's recommended for screening specimens. Midstream clean catch specimen. Specimens collected in a sterile container after cleansing the glans penis or urinary meatus. The first portion of urine is voided into the toilet. The mid portion is collected and the remaining portion is voided into the toilet. Nocturia. This is excessive urination during the night. Oliguria is a marked decrease in urine flow. Polyuria is a marked increase in urine flow. Suprapubic aspiration, the technique used to obtain sterile urine specimens for bacterial culture or cytologic examination, in which a sterile needle is introduced through the abdomen into the bladder. Timed specimen, urine specimen collected over an interval of time for a quantitative analysis of a urine chemical, usually a 24-hour collection. Questions. Question 1. Urea, creatinine, and uric acid are the three main blank chemical constituents found in urine. Choices: A, inorganic, B, organic, C, solute, D, formed element. The answer is B. Urea, creatinine, and uric acid are the three main organic chemical constituents found in urine. Question 2. The three main inorganic compounds found in urine are either A, S, P, and Cl, B, Ar, CH, and Na, C, Na, K, and Cl, D, Si, Cr, and Uc. The answer is C, sodium, potassium, and chloride, which are Na, K, and Cl. Question 3. I have a patient sample and I don't know if it's urine. If it is urine, it will have elevated levels of what two chemicals? A, creatinine and urea, B, sodium and potassium, C, nitrite and leukocyte esterase, D, silica and boric acid. The answer is A, creatinine and urea. Question 4. If a patient does not produce urine in a 24-hour, this is called A, anuria, B, oliguria, C, normal volume, D, polyuria? The answer is A, anuria. The patient did not produce any urine in 24 hours. Question 5. If a patient produces 1,250 milliliters of urine in a 24-hour period, this is A, anuria, B, oliguria, C, normal volume, D, polyuria. The answer is C. 1,250 milliliters of urine is a normal volume of urine. Question 6. If a patient produces 350 milliliters of urine in a 24-hour period, this is considered A, anuria, B, oliguria, C, normal volume, or D, polyuria. The answer is B, oliguria. This is a decreased volume of urine. Question 7. If a patient produces 2,560 milliliters of urine in a 24-hour period, this is A, anuria, B, oliguria, C, normal volume, or D, polyuria. The answer is D, polyuria. This is an increased volume of urine. Question 8. The urine collection cup should be A, wide-mouthed and flat-bottomed, B, narrow-mouthed and conical bottom, C, small and narrow, D, wide-mouthed and conical bottom. The answer is A, wide-mouthed and flat-bottomed. Question 9. Correct urine container labeling includes A, first and last name, label on the lid, B, first and last name, label on the cup, C, first and last name, date of birth, label on the lid, D, first and last name, date of birth, label on the cup. The answer is D, first and last name, date of birth, and the label goes on the cup. Question 10. The urine sample was collected at 1300 and brought to the lab at 1700. The specimen is A, acceptable as it is, B, rejected if stored at room temperature, C, rejected if stored in a refrigerator, D, accepted if stored in a freezer. The answer is B, rejected if stored at room temperature for 4 hours. Question 11. Which analyte increases in urine standing for more than 2 hours at room temperature? A, nitrite bacteria, pH and odor, B, nitrite bacteria, glucose and ketones, C, glucose, ketones, pH and odor, D, glucose clarity, urobilinogen, and bilirubin. The answer is A, nitrite bacteria, pH, and odor. Question 12. What action does gram-negative bacteria have on urine? A, it converts nitrite to nitrate. B, it converts bilirubin to urobilin. C, it reduces nitrates to nitrite. D, it increases glucose by glycolysis. The answer is C, it reduces nitrates to nitrites. Question 13. The most common preservative used for urine samples is A, boric acid, B, formalin, C, sodium fluoride, or D, refrigeration. The answer is D, refrigeration. Question 14. The most common sample type used for routine urinalysis is A, random sample, B, first morning, C, catheterized, or D, suprapubic. The answer is A, random sample. Question 15. The most common sample type used for culture. A. Random sample. B. First morning. C. Catheterized. Or D. Midstream clean catch. The answer is D. Midstream clean catch, which is more convenient for the patient and provides a good sample. Question 16. The sample that should have the least bacterial contamination and should grow no bacteria. A. Random sample. B. First morning. C. Catheterized. Or D. Midstream clean catch. The answer is C. Catheterized should have no contamination from bacteria, while midstream clean catch may have some, but it will be reduced compared to a random sample. Question 17. To avoid a false negative pregnancy test, use this sample. A. Random sample. B. First morning. C. Catheterized. D. Midstream clean catch. The answer is B. First morning, which is the most concentrated. Question 18. What sample type is used to detect a prostate infection? A. Random sample. B. First morning. C. 3 glass. D. Midstream clean catch. The answer is C. The 3 glass method. Question 19. For a 24-hour urine collection, which sample is discarded? A. The sample before starting the 24 hours. B. The last sample after 24 hours. C. The samples in the middle of the night. D. All should be kept. The answer is A. The sample before starting the 24 hours. You can see fadavis.com to find the book Urinalysis and Body Fluids by Susan King Strassinger and Marjorie Schwab de Lorenzo. Thank you for listening, and I hope this helps you learn more about urinalysis.